Hello, my name is Isabella Spielmann, and I will be telling you about my project, which is utilizing the CRISPR-Cas9 technology to knock down the phosphatase PRL3 and determine its role in pediatric medulloblastoma cells. I worked under the mentorship of Dr. Jessica Blackburn and with Caroline Smith, a PhD candidate in the lab. So for a basic outline, I will first provide you with my introduction and background information. I will then move into my purpose and hypothesis for my project. I will then discuss my methodology, my results and discussion, and then finish up with future directions, acknowledgements, reference, and then open the floor to any questions, or in this case, provide you with my email address should you wish to contact me about any questions. So to start, what is medulloblastoma? Medulloblastoma is a pediatric brain cancer that starts at the base of the brain and spreads upwards. Medulloblastomas make up about 20% of all malignant pediatric tumors, making them the second most common pediatric malignant tumor. Dowie cells are a group 3 medulloblastoma cell line. This means that they come from a medulloblastoma tumor, and group 3 means that they are a specific type of medulloblastoma. Medulloblastoma are categorized into 4 groups based on their cell signaling pathways. I chose this group 3 Dowie cell line because it contains my phosphatase of interest. A phosphatase works in conjunction with kinases for cell signaling or cell communication. In this image, the purple figure represents a kinase, and the blue figure represents a phosphatase. So in this image, a kinase in conjunction with ATP adds a phosphate group to a substrate. A phosphatase removes the phosphate group from the substrate. In this, these two molecules work to carry a signal through a cell. So why should we care about phosphatases? Phosphatases have not been widely studied as oncogenes, meaning they have not been looked at as possible cancer targets. The reasoning for this is because phosphatases are generally thought to turn cell signaling pathways off rather than on like their kinase counterparts. To date, there are no FDA-approved phosphatase inhibitors because they have not been widely studied. So the phosphatase of interest that I looked at was phosphatase PRL3, which is a part of the PRL family which contains 3 phosphatases. These PRLs are actually just proteins that act as phosphatases, but because the specific molecular pathway is unknown, the actual role of the phosphatase family, phosphatase PRL3 family, is also unknown. However, phosphatase PRL3 is linked to acute lymphoblastic leukemia. Phosphatase PRL3 is also overexpressed in medulloblastoma and Dowie cells, and it is linked to the progression of ALL or acute lymphoblastic leukemia. This means that PRL3 is partially responsible for the spread of ALL. So with these 2 pieces of information, the question was asked, is there a similar link between PRL3 and medulloblastoma as there is between ALL and PRL3? I hypothesized that PRL3 expression is essential for medulloblastoma viability, and I asked if PRL3 is necessary for Dowie cell growth and if it can be targeted using CRISPR-Cas9 technology. So for my methodology, I utilized CRISPR-Cas9 technology to knock down the PRL3 gene in Dowie medulloblastoma cells. I then used a genomic cleavage assay to verify my knockdown, and I used a cell titer glow assay to measure the cell viability of the cells after knockdown. So what is CRISPR-Cas9 technology? CRISPR-Cas9 technology is a gene editing technology that was discovered in 2011. The gene of interest is targeted by guide RNAs or gRNAs and bind to the DNA at these specified locations. The Cas9 protein then comes along and binds the DNA at the places marked by the gRNAs and acts like a scissors, cutting the DNA. There's then a blank in the DNA where mutations can be introduced. As the DNA tries to repair itself, it will make a mistake, causing mutation, which will render that gene non-functional. So the actual knockdown of the PRL3 gene using CRISPR-Cas9 technology was done in 3 steps. First, gRNAs, the first 4 targets were identified, meaning that there were 4 possible places where the cuts on the PRL3 gene could be made. So there were 4 gRNAs that were purchased. The gRNAs were then a virus was transfected with the gRNAs and then transduced onto Dowie-Cas9 cells so that the gRNA would actually enter the cells. So this was done by combining 2 viral packaging plasmids with the gRNA in order to make the virus. These were then applied to HEK cells, which acted like a factory producing lots and lots of the virus. Eventually, the HEK cells recognize that the virus is not a natural part of it and will kick the virus out into the media. At this point, the gRNA expressing virus will then be collected and transduced or added to target Dowie plus Cas9 cells. The gRNAs will then be pushed into the target cells with the help of the virus, allowing the Cas9 protein to then bind to the specified locations of the gRNAs that have entered the cells, and CRISPR should take place. After this was done, a verification of the CRISPR was needed. To do this, a genomic cleavage assay was used. In this, cells were lysed open to release the DNA, and then the specific section of where the mutation should have occurred were amplified, or a lot of it was made. The DNA was then denatured and reannealed, or it was pulled apart and then put back together. For this, half of the DNA could have had a mutation and half of it could not have. And so this DNA was mismatched so that at least one part of every DNA strand would have the mutation. A detection enzyme was then applied to make similar cuts to where the mutation should have been. And these were imaged on a 2% agarose gel. To measure the cell viability, a cell titer glow assay was used. In a cell titer glow assay, a reagent is applied to the cells and then should fluoresce in conjunction with the amount of ATP produced. So ATP is the standard, ATP production is the standard for which cell viability is measured, because if a cell is alive, it should be producing ATP. So did this work? This is the results of the genomic cleavage assay, or whether or not the PRL3 gene was able to be knocked down. In the first 2 columns, I have my negative and positive control that came with the genomic cleavage assay kit. These 2 columns represent what a good cut by the detection enzyme should have looked like. The negative control is without detection enzyme, and the positive control is with detection enzyme. So the negative sign always means without detection enzyme, and the positive sign means with detection enzyme. So as you can see with my control, in the yellow box, there's a distinct banding pattern of 2 horizontal lines with nothing in between them. These 2 lines represent a clean cut made by the detection enzyme. I ran my 4 target plasmids, or 4 target gRNAs, which are T1, T2, T3, and T4. And I additionally ran a scrambled plasmid, which should not have bound to the DNA, and regular Dowie cells without the Cas9 protein. The scrambled plasmid was a random plasmid that should not have bound to the PRL3 gene, meaning the Cas9 protein would have had nowhere to bind and no cut should have been made. This is reflected in the red box, as you can see a random banding pattern without the 2 clear, clean lines. Similarly, the Dowie cells without the Cas9 protein were run to verify that the Cas9 protein was actually doing its job of cutting. Because there was no Cas9 protein, no cut should have been made, and that is reflected inside the red box on the positive Dowie of another random banding pattern without those 2 clear cuts. For my targets 2 and 3 displayed the clear banding pattern of 2 clean cut lines that represent that the cuts have been made and that the PRL3 gene was knocked down. Unfortunately, for targets 1 and 4, they displayed a random banding pattern, meaning that the cuts were not successful and the gene was not knocked down. For the cell titer glow assay, the results of all of the targets were normalized to my scrambled. Along the y-axis, you can see the luminescence, or the amount that the cells fluoresced in conjunction with the amount of ATP. Along the x-axis, you can see my targets. I ran a one-way ANOVA test to compare all of my targets to the scrambled, and none of them displayed significant growth. So what can we conclude from this? There were several things to be concluded. First, the removal of the PRL3 gene is non-toxic to DAOI cells. This can be shown back in the cell titer glow assay results, as there was not a significant amount of growth or decay in any of the cells. This means that the cells can survive without PRL3. This is important because it could be potentially helpful in cancer treatments. Current cancer treatments for medulloblastoma include chemotherapy, which kill not only your target cells, but everything around them. Since PRL3 seems to be non-toxic, if that is a gene that can be knocked down that could slow the progression of medulloblastoma without killing everything around it, it would be extremely helpful in terms of cancer treatments. Additionally, the CRISPR-Cas9 system successfully knocked down PRL3 in targets 2 and 3. However, the degree to which PRL3 knockdown is unknown, meaning that maybe half of the gene was knocked down, maybe just a little bit was knocked down, and any remaining activity of the PRL3 gene could have an effect on the cell viability. Additionally, if this is not the case, another answer to why the cells remain the same is that there could be another phosphatase that stepped in to do PRL3's job. Since the specific signaling molecular pathway of PRL3 is unknown, we don't know how another phosphatase might act if it were to step in. For future directions, I would like to determine the level of PRL3 protein expression remaining after knockdown, meaning that the amount of the PRL3 that is left after knockdown is something to be explored. Determining if PRL3 is important for tumor migration is another step to take, and this can be done using a scratch assay or a transwell assay, meaning the amount that the tumor would move after the knockdown would be measured. These techniques can also be applied to other protein phosphatases. As mentioned previously, protein phosphatases have not been widely explored in terms of cancer treatment, and so these techniques could be used to further explore other phosphatases. I would like to thank my mentor, Dr. Jessica Blackburn, for letting me work in her lab and for guiding me through this process, as well as Caroline Smith for helping me along the way, the rest of the Blackburn lab for supporting me, MSCC, Ms. Karen Young, and KYRP for allowing me to have this opportunity. Here are my references. And if you have any questions, please contact me at this address. Thank you.